So a really big welcome to everybody joining this webinar. I'm going to pop my webcam on just for a second with my co-presenters as well, just so that you can see that we do really exist. This is not a recording. So I'm EyesOnALZ, and Ginger. Pietro, want to say hi? Brilliant. So I lead on an initiative called Cochrane Crowd, about which you're going to hear a lot more in a few minutes. I'm going to turn my webcam off now just so that I maximize the chances of good audio. And I'm joined today, as you've just heard, by two partners in crime, Pietro for Stall Catchers and Ginger for Mark2Cure. And they're both based in the US, and I'm based in the UK. So let me just pop my slides into presentation mode for the moment. I'm going to start off just with a little bit of housekeeping because some of you might be quite new to GoToWebinar. And I just want to briefly explain how you can interact with us, because we really do want to make this an interactive session and enable you to ask any questions that you might have. So you can ask questions in two main ways. You can either raise your hand, your digital hand, using the icon, the hand icon that should be next to your name in the list. And once you've raised your hand, we'll unmute you so that you can then verbally ask your question. You can also post a question in the questions box, which you should be able to see on the control panel quite far down. And we'll be keeping an eye on that questions box. And if it's a nice, quick, easy one of us might just try and answer it straight away, but we will try to summarize the questions that have been asked at the end. So in terms of structure, we're going to structure the webinar like this. We're going to first kick off with stall catchers. And Pietro is going to take us through the project and show us what you have to do. And we'll then have time for a couple of questions before moving on to Ginger, who's going to take us through Mark2Cure. And again, once Ginger's done, we'll spend 2 minutes on questions about Mark2Cure before moving on to Cochrane Crowd, which I'll take us through. I did then just want to spend a few minutes after that on the challenge week itself and just describing some of those logistics before hopefully having enough time for more questions and answers at the end. So just before I hand over to Pietro, I just want to take a quick poll. In GoToWebinar, you can set up polls, which is quite useful. And I just had one quick question. So I'm going to attempt to launch my poll. And so what you'll see in a minute is a question come up on the screen and some options. Just tick all the answers that apply. So this is really just to find out whether or not you've already taken part in any of these initiatives or if it's all completely new to you. So I've launched that poll. I can see some results coming in. That's fantastic. Brilliant. I think the numbers are slowing down now, so I'm going to close the poll and I'll just share those results with you. So really interesting to have this information. And it looks like, across the board, we've got some contributors who've taken part in each of the initiatives, but actually a significant proportion of you, 37%, haven't taken part in any, which is really useful to know and really good, to have you on this webinar. And I hope you'll get a really good sense of what all these initiatives are about and straight away afterwards go and dive into them, particularly Cochrane Crowd. I'm biased. Okay, so I'm going to close that poll and hide those results. And I'm now going to hand over to Pietro. So let me just try and make Pietro the presenter. Who is going to take us through stall catchers.
Hello.
Hello.
All right. So bear with me as I get this set up. Let's see how this looks. Are you seeing my slides?
Yep, we are.
Here? Okay, excellent. Okay, well, I'm. It's really great to be part of this this webinar and part of this this whole CitSci Bio Blitz. It's really exciting. I know it has a. This is, I think, the second year running, as you mentioned, and but the first year that Stall Catchers is involved. And part of what's so exciting to me is that you know, most of the citizen science that's done today is in support of conservation science, which is fantastic. But there's also this kind of narrow slice, which is health-based or medical-based citizen science. And it's very nice to see that we have you know, enough critical mass here to bring 3 such projects together into one event. So I think this is this feels sort of historic in some sense, and obviously as members of the human species, we all have some vested interest in our own health and medical science. So yeah, I think this is just a great activity, and thanks to Cochrane Crowd and to the Mark2Cure folks for inviting us to join in this. So so Stall Catchers is there's a schedule that I think you know, will be shared for the whole event, but our part is a 24-hour challenge, I think, as are the other portions, and it begins at 3 p.m. Greenwich Mean Time on February 26th. So you can sort of do your own time zone conversion for that. So what is Stall Catchers? The basic idea behind Stall Catchers is that you're helping to analyze Alzheimer's disease research data, and you do that. You can do that on a laptop or on a mobile device, and you basically see an outline of a blood vessel in a mouse brain, and you just have to make a simple binary decision. Either the blood vessel is flowing or it's stalled, and you have this little green slider that you move. And I'll give you a mini demo in a few minutes. But I think it's nice to look at the research that's compelling the citizen science project, because particularly in this case, it's very exciting research, and important discoveries have been made. So here you can see there's kind of this cycle being represented, and most people who are familiar with Alzheimer's disease associate it with these plaques that form in the brain. And when you do a post-mortem analysis on an Alzheimer's patient, that's actually how the diagnosis is made, to differentiate Alzheimer's from other forms of dementias if these plaques are present. But one aspect of the disease that hasn't been extensively understood or studied is that there's a reduced blood flow in the brain of Alzheimer's patients. Almost a 30% reduction in blood flow. So so our Cornell-based biomedical collaborators were investigating this, and they discovered that the reduced blood flow is due to these tiny little blood vessels in the brain called capillaries that are actually stalled for brief periods of time. But at any given moment in an Alzheimer's patient, there's a higher percentage of these capillary stalls than in a healthy person, and then when they looked at the downstream effects of these stalls, it indeed seemed to be causing this overall 30% reduction in brain blood flow. So then the question became, well, you know, what is the association between these plaques and the reduced blood flow? And it turns out that the disease model suggested by this, and their ongoing research seems to support this model, is that the amyloid plaques, which you see these little bundles at the top, when they're stuck together, they become toxic, and they cause an inflammatory process. So they not only kill brain cells, but they cause inflammation of the blood vessels in the brain, which is this next circle down on the right if you're going clockwise. And you can see those little stars are kind of that represents an inflammatory process, and it causes those capillaries to get stalled, and the blood stops flowing. And when the blood stops flowing, then the amyloid, the little blue things that stick together, which normally get cleared out by the vasculature, by the blood vessels, are no longer getting cleared out, and then you get more clumps. And
Pietro,
Yeah, yeah.
So sorry to interrupt. I just think we might have a bit of a stalling issue going on just with your slides because we're still just seeing the home page, the first slide at the moment, and I think you've probably moved on to the third or fourth.
Oh, okay. Yeah, let's check
Not
that.
sure why that is, but yeah,
Okay.
might just be
Um,
on a go slow.
so do you see a slide with a brain in the middle of it at this point?
No,
Okay. All right, let's see what's
I
going
did.
on. Okay. How about now?
No, I'm afraid I don't. Ginger, could you just confirm whether
Yeah,
you
I
can
just
see
see
the
your,
first screen, the first slide?
yeah.
Okay. Right, let me try a different approach here.
Ah, that worked. Something
Oh,
we saw the brain.
oh, you did see the brain. Okay,
We
so
did.
here,
There we go.
now do
Yeah,
We
you see
do.
the brain? But if I press play,
Brilliant.
do you still see the brain?
Yeah.
Oh,
yeah.
good. Okay. All right. So, um, yeah, sorry that wasn't there the whole time, but we'll, in the interest of time, we'll keep moving forward. So in any case, there's this promising body of work being done by Cornell that's very suggestive of a link between these stalled capillaries and the progression of Alzheimer's disease, and and the problem that the Cornell folks had is that going through and evaluating potential causes and treatments for these stalls required actually looking in the brains of these mice that have been given the human version of Alzheimer's disease and counting the individual stalled capillaries through these videos that have been taken at different layers of brain tissue. And in a single animal, we might be looking at thousands of these. So to answer one research question, to evaluate a single treatment, taking this very time-consuming approach could take 6 months or a year. And that was the impetus for creating this citizen science platform that would simultaneously speed up the research and inform people who are participating more about the disease itself and the potential role of capillary stalls in the disease. So, so the goal again was to sort of address this analytic bottleneck and reduce the time to find a treatment target from decades to potentially just a few years if we get enough people participating. And today we have about 8,000 participants. By the end of this year, we're hoping to grow our community to 30,000 participants and go from doubling the analytic rate to increasing it by a factor of 10. And we hope that you will help us do that through your participation in this event. So here, here are some basic instructions on how to participate in this. This is the
Yeah, your
um,
slides aren't moving on. I'm really sorry,
oh, okay,
Pietro.
okay.
I don't know
So
why. Oh, now they are. Yeah, you just have to click on it, I guess.
okay, and do you still see the slide now?
Yeah, we see the one with the different devices.
Okay,
Yeah.
so I think the easiest way is I'm just going to go ahead and walk you through
Oh, that'd be
getting
great.
started with it, and we'll just take a quick minute. So if you go to stallcatchers.com, this is the home screen you'll see, and there's this big bright Join Now button. So you click on that one, and we'll click on it again. There we go. Make up your own username. So and then you can put in your email address. So we'll say CitSci at crazyfun.com. We won't know if you put in a fake email address, but we hope you put in a real one. And if you do, then you get to go and adjust your password later if you'd like. I'm just going to go ahead and sign up. It's the clicking is I think affected by the webinar software. Here we go.
Mm-hmm.
Okay, so right away you're into the game here, and I'm going to skip the tour and just give you my own quick tour here. So what you're looking at is can you see this okay, Anna?
Yeah, it looks good.
Yeah.
Okay, great, thanks. So so this orange outline is showing you where to look as you move this green slider back and forth. And you can see that there's a white blood vessel that sort of follows the shape of the outline as you move it back and forth. And there's this dark gap in the vessel here that never seems to go away no matter where you move the slider. It just never gets filled in, and that's suggestive of a blockage or a stall. So then you click on the stalled button to say, I think that's stalled, and then click right where you think you see that. And hey, we got it right. So we caught the stall. There's a message to tell you a bit more about it when you're just getting started out, so you can understand, you know, if you just happen to get lucky, why was that the right answer? And then we move on to the next one. And so at each turn, you're doing exactly the same thing. So we have a new orange outline. And in this case, I don't know how well this comes across in the webinar, but you can see that there's movement, that these little black dots are actually flowing through the blood vessel. Does that come across, Anna?
Mm-hmm.
Yeah. And so in this case, we
Yeah,
call
really
it flowing. Good. Okay, great. And then move on. And there's really there's no way you can really mess up the data here, even if you tried. So making mistakes is part of doing this, and we combine lots of answers from lots of people to get one correct crowd answer. So please don't hesitate to play simply on the basis of, oh, I don't think I'm going to get it right, or I'm going to mess up the data. You really can't. We kind of you know, we built a lot into this to prevent those kinds of issues from being a factor. So in fact, I'll go ahead and I'll give a wrong answer on purpose here. Yeah. So just to show you, it's okay. You know, the system doesn't blow up and you don't get fired. So that's about it. I'm going to go quickly back to my slides that don't show up, and let's see. Can you see this
I
last
think
slide?
one suggestion is to not use the play function, and just if you've got your
Gotcha.
keyboard keys, I would try that.
Okay. Can you see this one with the links here?
Yes.
Yeah,
okay.
Brilliant.
Yeah, perfect. So you know, these are some quick links, and you know, we'll make these slides available after the webinar. So if you want to refer back to these links, it'll be easy to do that. And then again, here are the challenge dates for the stall catchers portion. And I think you know, Anna may mention also about how we're going to not only, you know, give awards for each individual you know, competition, but also have a kind of a combined award for folks who participate in all three and do well in all of them. So that's it for stall catchers. Yeah, thanks a lot, and good luck. I hope you have fun with it.
That's fantastic. Thank you. Shall we? We've got time, I think, for a couple of quick questions before we move on to Ginger. If people have a burning question, otherwise you can save it up for the Q&A at the end. So let's just have a quick check to see if any hands are up. So when I scroll down the attendee list, I know when you're an attendee in GoToWebinar, you don't know how many other people are attending. So just to let you all know, there's 52 attendees, which is really wonderful. No questions that I can see at the moment. So I think we should probably move on in the interest of time. So I'm going to now make Ginger the presenter.
All right. Let's see.
So in a moment, Ginger, you should get a prompt.
I got it. It's show my screen. Can you see it?
I can.
Okay.
That's great.
Can you see my slides?
We can see your slides, yes, in presentation mode. That's great.
Slide? Great. Perfect.
Yep.
All right.
Yep.
I'm going to, let's see. Let's try scrolling through here. Can you see? Did it change?
Yes. It's on a little bit of a delay, but it changed.
Okay, great. All right. So thank you, Pietro. That was good introduction. Thank you, Anna, for organizing this. And as always, it's really exciting to hear about the EyesOnALZ efforts because we love the idea that citizen scientists can help us learn more about diseases and ultimately how to treat them. And that's our hope for Mark2Cure. But for those of you who are not familiar with Mark2Cure, I'm just going to fill you in a little bit on how we got to where we are project. Mark2Cure is a project of the SU Lab at the Scripps Research Institute in La Jolla, California. Our lab is the computational biology lab, with a keen interest in making existing data information more accessible and useful. One approach we've been focused on is the crowdsourced approach. And a lot of members of our team in this lab actually work on enriching the biomedical content of Wikipedia and Wikidata and creating platforms for using the information from existing distributed and openly accessible resources. So our lab has built a number of tools for structured information, but a lot of incredibly valuable and important information exists in unstructured text. And the difference between structured and unstructured is really well illustrated by this Wikipedia article about NGLY1, which is a gene that was, this article was actually updated by an expert through the Gene Wiki review series. And you can see in the center, front and center here, this, this huge, easy to read block of text, which is an example of unstructured text. But also on the right, you can see this info box, which is an example of structured information. So you can see in front and center in the middle, this unstructured text can be broad, it can be highly variable, it can be deep, it can be nuanced. And so it's very hard for a machine to make heads or tails of it. But in contrast, the information in the info box here is organized, it's curated, and that's why it can be fed through templates into this info box, which can present it in a nice, neat visualization. So there's a lot of information that's organized in various databases, but a huge amount of information is not, and it simply exists as biomedical texts. And in biomedical texts, short of reading the papers, there aren't really that many ways to pull the information. So if you ask a researcher or a scientist what their typical day is like, a lot of them will tell you they spend a lot of time reading. But given that there are now over 26 million research papers and over 1 million new articles coming out each year, most researchers are only able to keep up with just a tiny sliver of what's already known. And many researchers are dealing with this dilemma where you can only read so much since you actually need time to do your experiments. So what if a drug or gene that's important to your domain of research was published outside of your domain of research? You wouldn't have been able to read about it or know about it. And our inability to keep up with literature is actually hampering our ability to understand how certain things are related. And these hidden unknown relationships could be really important clues for treating diseases. Mark2Cure is our attempt at asking volunteers to help us look at the literature and find how different genes, diseases, and treatments are related. Now, given the volume of literature, that's a lot to start with, especially since our project hasn't grown that much. We would need definitely a lot more users in order to cover 26 million articles or 1 million articles per year. And so we decided to limit the scope of the literature to focus our efforts. And we held a beta study 3 years ago with the idea that once we complete the beta study, we would focus the literature on the disease that was of most interest to people that helped us complete the beta study. So in our beta study, we demonstrated that non-experts could read and extract information from biomedical text, and that they were willing to do so, and they could do it really well. So if you'd like to read more about this study, it's open access. You need any sort of account to read it. And the DOI is right there in the bottom right-hand corner. And our beta study was completed thanks to over 200 citizen scientists and volunteers. But what was really neat was that there were members of different rare disease communities that helped us complete the beta study, especially the NGLY1 deficiency community. And they put together an impressively coordinated effort in order for us to be able to finish the beta study, leading us to launch our first campaign on NGLY1 deficiency. The scientific literature is hard to read, even for scientists. So how can non-scientists make a difference with our platform? First, because a lot of scientific literature is not openly available and is essentially pay-per-view content, Mark2Cure only deals with the portion of literature that is openly accessible, which is the abstract. Abstracts are generally written to be a little bit more accessible, so you shouldn't have to have a science background in order to read them. There's definitely a learning curve for reading abstracts, but reading is an instrumental way for people to learn and acquire knowledge. And we found that people who keep at it learn incredibly quickly. I've had the privilege of interacting with users who have, initially sending me questions, asking about the differences between a gene and a protein. And then they go on to debate with other users about whether or not protein isoforms or multi-protein complexes should be treated, like how they should be treated in the system. So in helping to investigate literature on this rare disease, you'll learn a lot about biomedical science straight from the source that we scientists get it from. And Mark2 curators have already become so good at this task. They've corrected errors in existing databases such as PubMed, which is a biomedical citation index. And they've also suggested entries for Drug Bank, which is a drug resource. And together, I think we can do a lot more. So what exactly are we asking you to do with these abstracts that you're reading? In Mark2Cure, we have two modules, one which we call entity recognition, where we ask you to help us find all the genes, treatments, and diseases in an abstract. And the other one is the relationship task, where we ask you to tell us how these different genes, diseases, and treatments are related based on an abstract. In the entity recognition task, volunteers are asked to highlight the genes, treatments, and diseases in different colors. And if you're worried about not knowing what a gene is or what exactly to look for, there's an online tutorial that teaches you what to look for in greater detail. But due to technical limitations, like this highlighting function doesn't really work well on a touchscreen device. This task should primarily be done on a computer or a laptop with a mouse because it makes it easier. Our other task, the relationship extraction task, can be done on a touchscreen or a mobile device. And with that, we essentially ask you to read the abstract and use the selection menu to report how different concepts are related. So far, volunteers have submitted over 1 million annotations revealing what genes, diseases, and treatments are in over 2,000 abstracts, and how these different genes, diseases, and treatments are related. And although our current campaign is limited to abstracts that relate in some way to NGLY1 deficiency, everything in the biomedical sciences is related in some fashion. So in joining the search for clues on NGLY1 deficiency, you may be uncovering clues in other diseases which may affect you in some way in the future. And so in the limited number of abstracts that we've actually annotated with Mark2Cure, a lot of diseases completely unrelated to, or seemingly unrelated to NGLY1 deficiency have been uncovered or extracted. So diseases that are common, such as diabetes and cardiomyopathy and obesity, have been covered in this curated set of abstracts. And rare diseases as well as Charcot-Marie-Tooth disease, autoleukodystrophy, Algor syndrome, muscular dystrophy, and more. And we all get sick at some point or another. And by contributing to Mark2Cure, you better prepare yourself to understanding the language of biomedical knowledge so that you won't feel like you're reading a foreign language for the first time when you're dealing with health issues. And this is actually the situation a lot of rare disease patients and families find themselves in. And in most cases, the most current source of information on their disease is the biomedical literature. But reading this literature for the first time can be really, really difficult. So if you can read, you can help. And we look forward to seeing you during CitSciMed Blitz.
Thanks, Ginger. That was good. We do seem to be having a few audio quality issues, but I hope people could hear Ginger okay. It just seemed at times a little bit garbled, but on the whole, it was okay for me. But I can see there are just a few comments. But if there are any questions for Ginger at this point, then we've got a couple of minutes now if we want to take those, or we can wait until the end. And can I just get a check just from people, is my audio quality all right? Or is it still...
It's good for the most part. There's occasionally like a little bit of like popping.
Yeah, yeah. I think we've just got some connectivity issues, haven't we? Well, Ginger, I think, thank you very much. We'll move on to the final presentation now, which is me. So I have to just grab the screen back from you. Let me just do that.
Great.
Show my screen and hop to the right slide. So hopefully you can see my presentation on the screen. Oops, maybe not. Now I've just done that.
Oh, yep, there you go.
Back again? Okay, wonderful.
Mm-hmm.
So I'm going to talk to you about Cochrane Crowd. But first, I wanted to just spend a minute or two telling you a little bit about Cochrane, just in case you're quite new to Cochrane. And Cochrane's logo, in fact, is a good starting point. What it shows is a forest plot with each of those horizontal lines representing a different study. And in this example, in this forest plot that forms Cochrane's logo, this was looking at studies that were measuring the effectiveness of corticosteroid therapy for fetal maturation. And that top horizontal line, that little one at the top, represents the first trial that was done in the 1970s. But it wasn't until over 20 years later, and all those other trials had been done, that the evidence was finally brought together by Cochrane, which came into existence then, and summarized. And the summary result is represented by that diamond at the bottom. And what it showed was that treatment was beneficial and that corticosteroids given to women who are about to give birth can save the life of the newborn child. And so that's basically what Cochrane does to this day, 20 years later. It summarizes the available evidence about treatments. The process is essentially this. It starts with a question. Does X improve Y for condition Z? That then kickstarts the search for evidence or the search for studies. And then once the studies have been found, along comes the synthesis process. I'm not quite sure why I visualized it as a giant purple Pac-Man, but that's just how I sort of see it at the moment. And it combines and summarizes all the evidence. So that all sounds quite sensible, I hope, but it's not without its challenges. And one of the biggest challenges, which Ginger has just alluded to, is information overload. There's just so much information being churned out, with global scientific output doubling every 9 years. And keeping up with all this information is a significant challenge for Cochrane, which is essentially an information brokering organization that tries to turn information into knowledge about treatments and whether they work. Cochrane's always needed and used volunteers to help produce good health evidence. But in the past, it has struggled to provide people with meaningful yet flexible ways to contribute. And it's, I think, fair to say it hasn't always catered well for those who are new to the world of health research. And so we built Cochrane Crowd, an online platform that hosts small, discrete tasks that people can dive into as and when they have time, helping us to keep up with the deluge of information. And so each of the tasks on Cochrane Crowd is about either helping to identify a particular type of research or helping to extract some details about it. And so this is a screenshot here of our very first and still ongoing task in Cochrane Crowd, which is called randomized trial identification. And here the citizen scientist is presented with a title and a summary or an abstract. And the question that we're asking is is this summary describing a randomized trial, or an RCT for short? And, not sure what a randomized trial is? Well, that's completely fine. All tasks are supported by training. And don't worry, it's not lengthy and boring training. It's brief, it's interactive, and it'll guide you through all that you need to know to do the task well. Because I think the biggest worry new contributors have is that of making a mistake. And that's where this thing called the agreement algorithm comes into play. And so this essentially means that no record is just viewed once. In fact, most records get 4 or 5 independent classifications. And this enables us to work out collective accuracy rather than base decisions just on individual classifications. So we've got our challenge coming up next week. Ours is taking place on the Friday, but I'll talk in more details about the timing of it. But I just want to say at this stage what you have to do to take part in the Cochrane Crowd part of the event. So the first thing you need to do is you need to sign up to Cochrane Crowd if you haven't done that already. And sign up is, of course, free and should take less than a minute. We're going to focus this challenge on a particular task in Cochrane Crowd, and the task is called CTID. And essentially, it's about identifying reports of randomized trials from the world's largest clinical trial registry called clinicaltrials.gov. And we do really need your help because we have around 20,000 records to get through. So no pressure, but we do really need some help. So step 2 is to do the training for CTID. If you want to take part, you can do it on the day during the challenge. That's absolutely fine. It takes around 20 minutes to do. So as I said, it takes you through some practice records. It's completely interactive, and it guides you through everything that you need to know for the task. And then, of course, the final thing is just at 3 p.m. when we kick off on the Friday, the 2nd of March. Log into Cochrane Crowd and you will see the challenge like that in your dashboard. So you'll know which task is the challenge task, because we have a number of tasks in Cochrane Crowd. And so I just mocked this one up today just to show you what it will look like, and it'll have the words challenge in progress across the top and then the name of the challenge. So in my last minute or two, I just want to describe 1 or 2 nifty features that are available in Crowd to help make the tasks more enjoyable or useful. And the first, and probably one of the most popular features, is that you can prioritize the records that you see in Crowd. So for example, if you have a particular interest in a certain health condition, you can go into the settings for that task, type in that condition, for example, asthma or diabetes or Alzheimer's, and then that will surface for you the records that are about those conditions. You can earn milestone badges. So these are visual reflections of your activity and performance within Crowd. A prestigious purple badge is very rare, but if you like a challenge, then it's one to aim for. And then for each task in Crowd, you can see how well you're doing under the History and Statistics tabs. So for example, and what this is showing is that I filtered by records where my decision or classification disagreed with the final classification given. And people have found this a really helpful way to learn when they might be making mistakes. So we launched Cochrane Crowd in May 2016. We now have over 8,300 contributors from 189 countries. And those amazing contributors have made over 1.5 million classifications and helped to identify almost 50,000 reports of randomized trials. We get lots of feedback, lots of suggestions, lots of really good criticism and suggestions for how we can make things better. And it's always great to hear comments like some of these ones here. So I like that I can make a small contribution to Cochrane from my own home on my own schedule. I hope to be able to contribute as much as I can in college and in my life after graduation, but I'd like to do something now. And when I saw that I can work directly with Cochrane, I was really excited. Feels like something useful you can do if you just have a few minutes and can't get into something bigger work-wise. It's great being a part of the team, and it's fun and actually useful. So with regards to Cochrane Crowd, I guess all that's left for me to say is if you haven't taken a look already, I hope you will go and have a look and consider signing up. And it would just be absolutely wonderful to have you all taking part on the challenge next week. And so with that, I'll come out of presentation mode and take a look and see if we've got any questions or comments at this stage before I talk a little bit more about the logistics of the challenge itself. And I do hope my audio was okay. Great, so let me just have a quick look at the attendees. If we don't have any questions at this point, I'll just move on. Oh, good, it sounds as though the audio was okay. So one question is where may I download the content of the webinar? So we're going to make the webinar available to everybody. We'll email around the link to the recording, but we'll also email around the slide sets as well if that's of interest to people, if they want to have those separately. So we've got about 15 minutes left, so I think I'm going to move on now, if that's okay, just to sort of final part. Oops, I've jumped to the end. Don't want to do that. Of this, which is just to briefly go through the timetable for next week, just to make sure everybody's clear. So as I'm sure you've grasped by now, we're going to run 3 24-hour challenges. We'll be kicking off with Stall Catchers first on the 26th of February at 3 p.m. UK time or GMT. We'll then have a little bit of recovery time, but not much, before the next challenge gets underway on the 28th of February, and again at the same time, 3 p.m. GMT. Before finally, last but not least, Cochrane Crowd Challenge starting on the Friday, 3 p.m. and finishing 24 hours later. And of course, we're all going to join in, and we're doing it to advance medical research and to play a role in helping deal with the significant challenges of information overload. But there will be prizes, and I'm sure that won't be the people's main motivation, but it helps, right? And what we're going to be doing is we're going to reward each individual within each project who does the best. So for example, if you screened the most records in the Cochrane Crowd Challenge, then you'll get your own trophy. And we're also going to reward the person overall who contributes to all 3 challenges and does, the most or performs really well across all 3 challenges. And this is just an example of what your trophy will look like, which I think is really, really nice. So that brings the presentations to a close. I'll pop back to questions mode here and go to webinar just to see whether or not we've got any more questions. I can see we have, so let me just open those out and have a look. So we've got, I'm a university librarian. Will there be things, tweets, links, whatever available that I can share easily with my students and encourage participation? Absolutely, that would be fantastic. So what we'll do is make sure that in the communication that we send around with the recording of this webinar, that we also include some bump about the challenge and the links to each of the 3 projects, because that would be absolutely brilliant. Yeah, can we share the webinar? Yes, please. Definitely. That would be wonderful. Next question. Can you briefly explain the Mark2Cure challenge? Yeah, absolutely. So Mark2Cure is it's an annotation task, And Ginger, shall we just try your audio again if you want to chip
sure.
in?
Can you hear me?
Actually, yeah, that sounded good to me. Yep.
Okay, so basically, you will be doing either module is fine. It's just pretty much doing annotation tasks, either for the entity recognition or the relationship extraction.
That's great. And can I just ask, Ginger, in terms of, training, because I sort of think maybe the Mark2Cure challenge out of all three is probably the most challenging to some extent.
Yes.
If you wanted to sort of give advice on which one was the easiest out of those two, entity recognition or identifying the relationships, which would you say?
That depends. So our user base have given feedback pretty much depending on whichever one they trained on first. So the users who started with entity recognition like entity recognition, they want to stick with entity recognition. They think entity recognition is the easiest, but the users who started with relationship extraction.
That's interesting.
Yeah. So it's what you get used to, I think.
Yep. I often get asked a similar question with Crowd because we have tasks that are about identifying different types of studies, and then tasks that do come across as being more in depth that are about describing those studies. But actually, we're finding that an awful lot of people quite like the more in-depth describing, even though it might take slightly longer. They actually feel it's less challenging because if something's well reported, you can extract the stuff you need reasonably easily, like the number of participants in a study. If something's really badly reported, then there's just an option to say, well, it's not reported or it's not clear, so you can then move on. So I'm getting quite a lot of questions now, so let me just move on and see what else we've got. So would I be informed about my work kind of correct or incorrect? And I think that's a really good question and probably one for all three of us to answer. I'll just go ahead and answer from the Cochrane Crowd perspective. The brief answer is yes. As I showed you just briefly in the slides, in Cochrane Crowd, we do try and have feedback mechanisms in place, automated feedback mechanisms in place that help you to see how you're doing performance-wise. And so what you can do is look in your statistics tab or in your history tab, and you can see the number of records and the records themselves that you kind of got wrong, if you like, and maybe then understand a little bit more about where you might be going wrong. But we can also convert, depending on how many you do during the challenge, we can turn that into an accuracy percentage. It only really becomes meaningful once you've done a reasonable number of records because of the prevalence of RCTs being reasonably low. But yeah, certainly I think we would hope to show you some performance data, and the rewards at the end will be a combination of quantity and performance metrics. Ginger, do you want to go ahead and talk briefly about?
So we won't have an absolute correct or incorrect for you. Instead, we will, usually in our feedback screen, we'll pair you, if you're in entity recognition, we'll pair you with the user who has done the best for that particular abstract, and you can compare your annotations with theirs, and you'll be provided with a score which is based on how you compared with the crowd, essentially.
That's good. Pietro, any comments?
Yeah, sure. So in Stall Catchers, we have two types of questions that we give. One's where we already know the answer, in which case we can evaluate your answer and tell you if it's correct or incorrect. And we use those to sort of track your progress and then appropriately sort of calibrate your answers as we combine them with answers from other people. Then we also have what we call the maybes, our true research vessels, where we don't have an answer. We're seeking an answer from volunteers. And in those cases, we can't say if you're correct or incorrect because we don't know, but we say maybe, and then we will keep track of the ones you've answered, and later when you come back, if we've accumulated enough answers from other people to have a crowd-based answer, then you will get to redeem points if you're in agreement with a crowd answer. So we have an actual button called redeem, and when that button lights up, it means that you actually have that opportunity to get some points for some of the maybes you've answered. So for the first kind, the calibration vessels where we know the answer, if you get it right, you may get a lot of points right away. With the maybes, you'll get a small number of points, and then later, if you agree with the other volunteers, you'll get a much larger number of points.
That's brilliant. Thank you. I'm just going to read out one of the questions we've had, even though it's been answered, just in case other people aren't watching the typed questions. So is there any special links for the challenge for Stall Catchers and Mark2Cure, like there is in Cochrane Crowd? You'll be able to see a particular task that's a challenge mode. So no, you just go to Mark2Cure and Stall Catchers when the challenge is running and take part. There isn't a special, entry into it or a big sort of flashing sign about it. And one other question.
I would add.
Oh yeah, go ahead.
Sorry, I was just going to add that the stall catchers during that period of time will be running in a special challenge mode. So there will be a challenge leaderboard, which is specific to the challenge and sort of keeps track of people in terms of their progress, and is ultimately what would guide the awards from our standpoint. So yeah, I think you'll see that there's a challenge going on, and I think we're actually doubling points. So this actually gets our existing community quite excited when we have events like this, because there's this opportunity to suddenly get twice as many points for your effort.
That does sound good. One other quick question I think somebody asked a bit earlier on, I'm just trying to find it, was what time the Mark2Cure challenge gets underway. So I don't know if on my screen you can see my slides. The Mark2Cure one starts on the 28th of February at 3, and they all start at the same time, 3 p.m. GMT. Oh yeah, that's great. So we are just about on the hour. Let me just have a quick look to see if we've got any hands raised. Nope. What I'll do is after this, collate any of the questions as well, so that we've got answers to those, and put that in the email as well, so that if you do share with other people and they have similar questions, hopefully we've answered those. If you do think of any other questions, then please do just get in touch. I haven't put our contacts on the screen, but let me just see if I can do that.
So Anna, while you're doing that, I thought I would just mention briefly that that we'll be using a hashtag for the entire event, I think, which is CitSciMed Blitz. I think I posted it as an answer to one of the questions, but what we'll try to do is post some links also on Twitter at that hashtag. So, if you walk away from this webinar and you're thinking, where do I go next to, download the webinar, get links to the individual projects, and so on. If you just remember the CitSciMed Blitz hashtag and you search that on Twitter, we'll I'll be posting relevant content and information, and also tracking the progress of the challenge using that hashtag. So it's just a way to kind of stay in the loop. Yeah,
no, that's brilliant. Thanks for mentioning that. I had forgotten about that. I don't seem to have my email anywhere here, so I'm just going to say it out loud. It's just simply crowd@cochrane.org. So, and I'm happy to field any questions, even if they're about stall catchers or Mark2Cure. I'll pass them on to Ginger and Pietro. So that's just crowd@cochrane.org. And thank you very much, Mary, for your comment. Yeah, I hope this is fruitful for you all. So exciting. Good luck. Yeah, now that we're doing this webinar, I'm really feeling excited about this and raring to go. So this time next week, we'll have just got Mark2Cure challenge underway, having done stall catchers, and Cochrane Crowd will still be waiting to go. It's going to be good. It was really good fun last year when we did it with Mark2Cure, and I think we just wanted to build on that, didn't we, Ginger? And stall catchers just looks like such a good, fun, really, brilliant citizen science task. So I'm definitely going to be trying to get my double points next week.
I'll be watching to make sure you do, Anna.
I want that trophy, to be honest. It looks fantastic. I'm quite competitive. So I just want to say a huge thank you to everybody for attending. We're on the hour now, so I'm going to draw the webinar to a close, but it's been really, really good and great questions. So yeah, good luck to everybody next week, and let's all just stay in touch via Twitter and email us if you have any other questions, and we'll post around the recording of this webinar. So thank you very much, and goodbye from me.
That's great. Thanks so much. Yeah, this has been really great. Thanks to everyone for showing up and listening in on this. Hope you all have fun with it.
Thank you. Bye.
Thank you. Bye.